Okay, so last time we saw how the light-dependent reactions produce NADPH and ATP by converting the energy in light first into a electrochemical potential and then later into ATP through chemiosmosis. So that's shown here in this slide on the left side of the slide, where we have the oxidation of water that's driven by light, leading to a buildup of protons inside the lumen of the thylakoid. We're going to make some NADPH when those electrons that came from water and were photooxidized get deposited onto NADPH with the enzyme help of NADP reductase. And then we're going to use the proton gradient to drive the synthesis of ATP. In the light-independent reactions, which are shown here as labeled as the Calvin cycle on the right of this slide, we're going to use that NADPH and ATP to accomplish really three major things. We're going to first fix carbon out of the atmosphere using a very abundant enzyme called Rubisco to join a molecule of carbon dioxide onto one molecule of a 5-carbon substrate. That is the carbon fixation reactions, or the carboxylation step, shown here as stage 1. That's going to produce a small organic molecule that's not yet a true carbohydrate, and that's where our ATP and NADPHs come in. We're going to reduce that small organic 3-carbon skeleton by adding electrons from NADPH. And when that reduction and phosphorylation using our ATPs is accomplished, we're going to produce a true carbohydrate that's only 3 carbons large. It's called glyceraldehyde 3-phosphate. And that's the second step. That's the reduction step. That is a series of enzyme-mediated reactions. It's shown here as a single step or maybe a couple, but just for simplicity's sake, we've left a bunch of intermediate steps out. I want you to focus on the idea of 3 major steps: carboxylation, reduction, and then regeneration, which I'll come to a little bit later and I'll describe in a minute. So let's talk in a little more detail about the carboxylation reaction. This is, again, joining a carbon dioxide from the atmosphere onto an already existing substrate in the Calvin cycle, again, a metabolic cycle, and that already existing substrate is a 5-carbon, really highly reactive molecule that receives the carbon dioxide. The name of that carbohydrate is shown here, ribulose 1,5-bisphosphate, and that's where the name of the enzyme comes from. Rubisco can carboxylate ribulose 1,5-bisphosphate. The 1,5 refers to the positions of the phosphates, and so we know based on that name that the phosphates are on the ends of the 5-carbon substrate, and that's part of what makes it so reactive. This enzyme Rubisco is special. It's the most abundant enzyme on the planet. This is where all of the carbon fixation occurs. This enzyme is doing all carbon fixation. So all of our reduced carbon, which includes all of the known fossil fuels, natural gas, oil, coal, all of that was fixed by the activity of Rubisco some 250 million plus years ago, which is kind of fun to think about. So this is our first phase of the reaction, and this is really only a single enzyme-mediated step, the carboxylation step. It's not using any energy directly, but because, as you can see at the top of this diagram again, it's taking a highly energetic molecule, ribulose bisphosphate, and that was really formed by the investment of energy from the light reactions. But the Rubisco enzyme-mediated step is not directly requiring any additional energy input. The result of that Rubisco carboxylation reaction is phosphoglycerate, PGA, as it's labeled here. And it's a simple 3-carbon organic molecule. It's got 1 phosphate on it. So really, when we carboxylate RUBP, we end up with 2 molecules of 3-phosphoglycerate. And those molecules of phosphoglycerate are then phosphorylated a second time, so they're the other end of the carbon skeleton, receives a phosphate group. And that, again, keeping with this principle of phosphorylation makes things more reactive, that investment by the cell of ATPs that, keep in mind, were made over in the light-dependent reactions, that investment makes 1,3-bisphosphoglycerate, and that can then be reduced by NADPH. Those electrons, remember, on NADPH, where did they come from? They came from water, ultimately. So that reduction reaction on phosphoglycerate or on 1,3-bisphosphoglycerate produces a small carbohydrate called glyceraldehyde 3-phosphate. It's a 3-carbon carbohydrate. It's reduced enough to qualify as a carbohydrate, and it is really the product, the exported product of the light-independent reactions. We think of sugar being the main product, but this is the form that the Calvin cycle releases product to be converted into sucrose or to be made into starch in the cell. So this is known as the reduction phase or reduction step. It's actually a couple of enzyme-mediated reactions. We're going to use that ATP first to phosphorylate phosphoglycerate, make it more reactive, so it's a priming reaction. And then that 1,3-bisphosphoglycerate is reduced to glyceraldehyde 3-phosphate. The reduction, the electrons to carry out that reduction come from NADPH. So that's where all of the NADPH that's generated by the light-dependent reactions, that's where it's used. The final step is to regenerate, because we're a metabolic cycle here. The Calvin cycle regenerates the initial substrate, that 5-carbon compound ribulose bisphosphate. And so all of the rest of the steps, which are numerous and they're not shown here, fairly complex, but we're going to take that 3-carbon glyceraldehyde 3-phosphate, and through a series of a lot of chemical transformations, that we're going to turn it back into the 5-carbon substrate for the entire cycle that can be carboxylated again by Rubisco and go through the whole cycle. So that phase is called the regeneration part of the pathway. We're regenerating the initial substrate. And again, a little bit of energy is required to do this. It's ATP. This slide says NADPH2, but I got carried away. It should just say ATP from the light reactions, and just a little bit, just three more ATPs. And so altogether, if we go back to our cycle slide, altogether, what we see is to produce 1 molecule of glyceraldehyde 3-phosphate. That's a 3-carbon molecule. And so we really have to turn the Calvin cycle 3 different times. To produce 1 molecule of glyceraldehyde 3-phosphate requires the incorporation of 3 carbon dioxides, and that requires the input of 9 ATPs altogether, and 6 NADPHs. So keep that in mind. 1 molecule of glyceraldehyde 3-phosphate requires 9 ATPs and 6 NADPHs, and of course the incorporation of 3 molecules of carbon dioxide. So what you can see here is that maintains balance for the regeneration part of the pathway. We send back in, or we maintain 5 molecules of glyceraldehyde 3-phosphate for every 1 molecule we want to export from the pathway. So when we think overall about the light independent or the carbon reactions, the main product of the carbon reactions is glyceraldehyde 3-phosphate. That's the main source of carbon output from the cycle. And this stuff, it can remain in the plastid. And in that case, if it stays inside the plastid, it enters the starch biosynthesis pathway for storage as starch in the plastid. When it's exported from the plastid, it can enter the sucrose biosynthesis pathway, and in that case, it's going to be leaving the cell for transport somewhere else in the plant. So again, the net incorporation of 1 of these 3-carbon carbohydrates requires 3 carbon dioxides, 9 ATPs, and 6 NADPHs. Those ATPs and NADPHs, where did they come from? They came from the light-dependent reactions. So you can see the carbon reactions absolutely rely on the light-dependent reactions for the input of energy, which ultimately came from light.